Thank you all for coming to this talk, exploring associations between cancer genes and pharmacological compounds, target drug networks, and GEDA. My name is Monica Arroyo, and I am an Associate Professor of Chemistry at Pontifical Catholic University, Puerto Rico. Before I begin, I would like to thank the organizing committee for tirelessly working to put together this international symposium amid a pandemic. While it is unfortunate that we cannot come together in person, I am most honored to be invited to participate and offer you what I hope is an interesting and motivating talk. The National Institute of General Medical Sciences, NIGMS, defines pharmacogenomics as a field of research that studies how a person's genes affect how he or she responds to medications. Its long-term goal is to help physicians select the drugs and doses best suited for each person. Thus, pharmacogenomics is part of the field of precision medicine, which aims to treat each patient individually. Altman and colleagues said it well in 2018, that the field of pharmacogenomics seeks to elucidate the interactions between genetic variation and the metabolism mechanism of pharmaceutical compounds. So why are we interested in pharmacogenomics? Loverty and colleagues pointed out that the prevalence of prescription drugs highlights the need for a better understanding of drug response, especially with regards to drug-gene, drug-drug, and drug-drug-gene interactions, and that the goal of pharmacogenomics research is to further the understanding of these interactions. In 2020, an estimated 1,806,590 cases of cancer will be diagnosed in the United States, and 606,520 people will die from the disease. That's on top of COVID-19. About 1.3 million new cases and 666,000 cancer deaths were estimated to have occurred in 2018 in Latin America and the Caribbean alone. In fact, in 2018, there were 18.1 million new cases and 9.5 million cancer-related deaths worldwide. Needless to say, cancer is among the leading causes of death worldwide. We all know someone with cancer. Perhaps we ourselves or a relative have fought the disease. Perhaps we have lost someone to it. Unfortunately, cancer spares no one. Despite all the recent advances in cancer research therapies and medical technologies, much more needs to be done. Here you can see the cancer age standardized rate per 100,000 people. Please keep in mind that standardization is necessary when comparing populations that differ with respect to age, because age has a powerful influence on the risk of dying from cancer. The World Cancer Research Fund has the global cancer rates for both sexes, focusing in the top 50 countries. The countries in the top 12 come from Oceania, Europe, and North America. As you can see, Australia is number 1 in the world, while the USA is number 5. Japan is the only Asian country in the top 50 at number 43. Spain is number 29. Uruguay, the symposium host country last year, is at 31. And Puerto Rico, where I live, at number 36. Israel rounds up the top 50. I should point out that Uruguay, French Guadeloupe, and Puerto Rico are the only Latin American Caribbean countries in the top 50. When we look at these stats a little deeper, we find a trend. The cancer most prevalent in the vast majority of South American countries is lung cancer. The western portion of the continent is most affected by stomach cancer. Bolivia has a prevalence of gallbladder cancer, and Guyana, cervical cancer. Central America presents quite a variation, while Mexico and Puerto Rico present mostly cases of colorectal cancer. When we look worldwide, as of 2018, the most common causes of cancer deaths are lung, with 1.76 million casualties, followed by colorectal, stomach, liver, and breast cancer. As you can see, the top 4 are also common in Latin America. So giving up is not an option. We must continue to research the molecular intricacies of cancer, as well as seek new treatment therapies. And that's where we come in. Using bioinformatic techniques and pharmacogenomic approaches, we are looking for new plausible therapies to identify new molecular targets to help curb the ravages of cancer. Cancer is a complex disease where multiple genetic changes rewire cellular networks during carcinogenesis. This indicates that cancer drug therapy needs the implementation of network-driven studies to reveal multiple interactions between cancer genes and drugs. As you all know, this carries an intrinsic complexity at the moment of carrying out research. Now, Albert Einstein said, if we knew what we were doing, it wouldn't be called research, would it? Those are words to keep in mind when we feel overwhelmed and perhaps even confused, as we set about to do research, explore new methods, and contribute to knowledge, regardless of the system we're studying or where our interests lie. So where do we even begin? There are between 40,000 and 600,000 predicted PPIs in the human interactome. PPIs are protein-protein interactions. Targeting cancer protein-protein interactions is paramount since its capacity to interact with a protein-binding partner is a direct consequence of the tumor-promoting character of many proteins that are expressed abnormally in cancerous conditions. For this, we first explore primary protein-protein databases, which contain data curated directly from the scientific literature. There are many types of databases. As you can see here, of particular importance are INTACT, MINT, DIP, and BioGrid. INTACT, MINT, and DIP are generalist databases. MINT has the largest number of organisms at 647. INTACT has well over 1 million interactions available. BioGrid focuses on model organisms and humans. These databases have thousands of proteins and interactions, with some overlap between them. We next have secondary or meta databases, which integrate and unify primary protein-protein interaction databases. These databases not only extract data from PPI databases, but also from other kinds of databases to provide supporting information. APID and PINA, for example, are traditional PPI metamining databases since they periodically download data from source PPI databases to create a centralized repository. Mentha is another important database not shown in this figure, and it contains almost 100,000 proteins and almost 750,000 PPIs. I would like to highlight that APID was developed by Diego Alonso and Javier de las Rivas at University of Salamanca, and it provides a comprehensive and curated collection of interactomes for more than 1,100 organisms, derived from the integration of experimentally detected protein-to-protein physical interactions. In fact, APID includes 90,379 distinct proteins and 678,441 interactions. So far, I have shown you a very succinct overview of PPI databases. But what about those for cancer? The fact is that there is a significant number of cancer databases and resources, but there are a few that I would like to briefly show here. The National Cancer Institute Genomic Data Commons (GDC) data portal provides the cancer research community with a unified data repository that enables data sharing across cancer genomic studies in support of precision medicine. The portal is a robust data-driven platform, an interactive system that allows cancer researchers and bioinformaticians to search and download cancer data for analysis. As you can see, it contains data from 67 cancer projects, over 80,000 cases, and over 3 million mutations. Over in Europe, at the Wellcome Sanger Trust in the UK, is COSMIC, the Catalogue of Somatic Mutations in Cancer. This is the world's largest source of expert, manually curated somatic mutation information relating to human cancers. COSMIC has data from over 37,000 genomes from peer-reviewed large-scale genome screening data and from other databases, such as NIH's The Cancer Genome Atlas program. There are two more databases I'd like to mention before I weary you. There is OncoPPI, which is a high-throughput screening platform that integrates the network of experimentally detected cancer-associated PPIs with cancer genomic, pharmacological, and protein structural data. OncoPPI is a relative newcomer, set up in 2018, and it limits the PPIs reported to physical association. OncoPPI has a total of 364 PPIs that were extracted from multiple databases, including BioGrid and IntAct. Now, though not dedicated to cancer, another important database is STITCH. STITCH is a massive database of known and predicted interactions between chemicals and proteins. The interactions include direct, physical, and indirect functional associations from computational prediction, knowledge transfer between organisms, and from interactions aggregated from other primary databases. As you can see, there are many databases to choose from. This is both a good thing and a problem. It is a good thing because there are many resources available with different kinds of information and sources to perform bioinformatics research. On the other hand, because there isn't a central database or repository with a defined and unifying format, a researcher can spend a significant amount of time researching from database to database and not necessarily find what they need. Moreover, not all databases allow researchers to download the full dataset for further exploration, thus limiting research efforts. Improvements in survival has been driven primarily by progress in early diagnosis and new drug therapies. Survival, unfortunately, is still profoundly affected by tumor metastasis and drug resistance. Therefore, the identification of the molecular targets of each drug and the discovery of new drug targets in cancer are essential to improve the effectiveness of tumor therapy. Now, in late 2014, I applied to Banco Santander's Fellowship for Young Researching Professors. I initially refused to apply because I was slightly over the age limit, but a colleague of mine kept insisting daily that I should apply, and so I did. And that's how I went from my university in Puerto Rico to University of Salamanca in Spain to work in the Bioinformatics and Functional Genomics Research Group of Dr. Javier de las Rivas in the Cancer Research Center in the summer of 2015. What began as a 10-week research exchange experience became a collaboration with a yearly summer visit, except 2020, of course, because of the pandemic and travel restriction. Thanks, COVID. Now, the beauty of this lab and collaboration is that everyone has a different background and experiences, bringing their own strengths to the various research projects taking place. As such, our pharmacogenomics research interests include identifying potential protein targets of drugs without known molecular targets, identifying new targets of approved drugs, using drug repositioning to identify new therapeutic uses for approved drugs, determining protein targets that induce drug resistance, and exploring drug target associations that may be relevant to diseases other than cancer. So, how do we go about these interests and goals? Like most of you, we take publicly available data, develop scripts, and perform multiple analyses to identify new targets and potential drugs related to cancer. Easy peasy, right? Over time, we have performed a large-scale screening to find associations of drugs approved by the FDA for cancer treatment, as well as small molecules approved for other diseases or no condition at all, with human genes. We use the standardized expression for 705 genes versus the standardized activity for over 20,000 compounds, and calculate the Pearson and Spearman correlations for all possible pairwise gene-drug combinations, which we then narrowed to the FDA-approved cancer drugs. These correlations were then used to build a global bipartite network that includes 1,007 gene-drug significant associations. The data were then integrated into an open web tool called GEDA, Gene Expression and Drug Activity, which includes a relational view of cancer drugs and genes, disclosing the putative indirect interactions found for these FDA-approved drugs, as well as the known targets for these drugs. Fundamental to our studies have been both CellMiner, COSMIC, which I previously mentioned, and the Genomics of Drug Sensitivity in Cancer GDSC databases. Data about the genome-wide expression of human protein-coding genes in the ACN60 cancer cell lines were downloaded from CellMiner, a pharmacogenomic resource that reports the activity of over 20,000 compounds. It also contains information about the activity or expression of over 22,000 human genes, as well as quantitative profiles of 10,350 NCI60 cancer cell lines. The expression data from the human genes corresponded to mRNA normalized signals for each gene, converted to average z-scores. The selection of cancer genes used in our studies was taken from the Cancer Gene Census (CGC) dataset from the COSMIC database. A set of 705 cancer-related genes, common between CGC and cell minor, was analyzed, while the GDSC was used to corroborate some results. This figure is from our most recent publication in Biomolecules, and it shows a comparison of gene expression versus drug activity, normalized profiles, to find significant correlations. In panel A, we have the EGFR gene expression versus dasatinib's drug activity. Dasatinib is a BCR-ABL1 tyrosine kinase inhibitor used in the treatment of Philadelphia chromosome-positive adult acute lymphoblastic leukemia and chronic myelogenous leukemia. The bar plots show that EGFR is expressed in lung, breast, ovarian, renal, and some colon cancer cell lines. In panel B, we show the positive significant correlations of dasatinib with 12 oncogenes. Some of these genes have been previously identified as drug targets, while the others are proposed as potential new candidate targets. EGFR is not a known target of dasatinib, but our analysis found this pair to have a significant correlation. Dasatinib has two known targets, ABL1 and PDGFRB, that were also found in our analysis. In our literature search, we found several studies that reveal the action of dasatinib on EGFR, which support our results that the correlation identified between dasatinib and EGFR could reveal an effective association. Here we show a bipartite-directed subnetwork of 4 drugs, afatinib, dasatinib, erlotinib, and ibrutinib, and their target cancer genes, 25 oncogenes in blue, 15 tumor suppressor genes in green, and 10 other cancer-related genes in gray. The links, the gray arrows, show the significant correlation found between the expression profiles of the genes and the activity of the drugs. Our results indicate that the activity of 3 of these drugs presents a positive correlation with the expression of EGFR, which is especially significant in lung and renal cancer. This observation is supported by a clinical trial published in 2014 by Gold and co-workers, in which a combination of dasatinib and erlotinib was successful in the treatment of non-small cell lung cancer, NSCLC, in patients with activating EGFR mutations. This final figure from our paper presents significant correlations found for 2 drugs that are frequently used to treat hematologic malignancies, venetoclax and cyclophosphamide. Venetoclax is a BCL2 selective inhibitor for the treatment of relapsed chronic lymphocytic leukemia and acute myeloid leukemia. Cyclophosphamide is a cancer spectrum drug that has been extensively used for the treatment of various types of lymphoma and leukemia. The bipartite-directed subnetwork of venetoclax and cyclophosphamide show 13 oncogenes in blue, 8 tumor suppressor genes in green, and 3 other cancer-related genes in gray with unassigned roles. Given that our results show that venetoclax and cyclophosphamide share a common gene, BTK, we decided to explore if these 2 drugs have been used in combination therapy. Indeed, at present, there is a clinical phase 1-2 trial to study the effect of these 2 drugs in patients with ALL. Venetoclax is a BH3 mimetic that specifically inhibits the anti-apoptotic BCL2, causing apoptosis of CLL cells. Yosifov and colleagues showed recent advances in CLL therapies, including new combination schemes and novel BTK inhibitors. The drugs in blue are not currently approved for CLL treatment, while those in black are in various stages of clinical development. This work supports our current studies, in which we are currently carrying out to further analysis on our datasets, focusing on compounds not approved by the FDA for cancer therapies and compounds that, at the moment, are not approved for any therapies. I don't have time, unfortunately, to go over our findings in detail, so please allow me to read this excerpt from our most recent paper. Our results show plausible or expected cellular targets for FDA-approved cancer drugs, as well as new candidates derived from a thorough analysis of gene expression and drug activity. As such, our study may contribute to a better understanding of the mechanism of action of many drugs and present an alternative to exploring drug side effects and drug resistance. Finally, the development of GEDA may help researchers in identifying significant novel interactions and cancer gene drug clusters to better understand the underlying molecular mechanisms of cancer therapy and drug action. Before I wrap this up, I'd like to invite all of you to see Alberto Barral Gonzalez's poster and flash presentation titled Bipartite Networks of Cancer Genes Associated with Specific Drugs Using Pharmacogenomics. Alberto is an extremely talented PhD student in Dr. Javier de la Rivas' lab at University of Salamanca. He will be showcasing GEDA and some new and exciting results, so don't miss his presentation. Now, I would like to thank Dr. Javier de las Rivas and all the members of Lab 19 at University of Salamanca's Center for Cancer Research for the collaboration, friendship, and support. In addition to Javier, I'd like to especially thank Alberto Barral Gonzalez, Diego Alonso Lopez, and Santiago Buenafuertes, who have worked tirelessly in this investigation, as well as Francisco Campos Laborie, a former PhD student, now a research associate at the Wellcome Trust Cancer Research UK Gurdon Institute at University of Cambridge. We have 2 papers published cited here, and we're currently working on another one. Friedrich Gowland Hopkins, Nobel Prize winner and biochemist, discoverer of vitamins, said this phrase that has a ring similar to Jane Austen's famous line in Pride and Prejudice. It is an old saying, abundantly justified, that where sciences meet, there growth occurs. It is my hope that as we come together in this conference from different countries and interests to share our research findings, we have a good time despite the distance and circumstances, and we can form lasting bonds. Thank you for your attention and enjoy the symposium.